Hello, my name is Natalie Usher, and today I'm going to be discussing the regulation of sperm maturation in the germinal epithelium. So first, let's discuss exactly what the germinal epithelium is. And I have a little diagram here to kind of represent it. The germinal epithelium is the innermost layer of the testicle. The germinal epithelium can also be known as the wall of the seminiferous tubule within the testes. The cells in the epithelium are connected via tight junctions. And there are two very different types of cells contained in the germinal epithelium. The first of these is the spermatogenic cells. And these develop to eventually become sperm cells. Typically, these spermogenic cells will make 4 to 8 layers inside of the germinal epithelium. The other type of cell contained in the epithelium are Sertoli cells. The large Sertoli cells, which are not dividing, function as supportive cells to these developing sperm. So, the development of a mature sperm cell takes place in 2 steps: spermatogenesis and spermiogenesis. Spermatogenesis, which we're going to talk about here, is the process by which a spermatogonium develops into a spermatid. During this process, the number of chromosomes is halved through meiosis, and it involves the following stages. Spermatogonia rest on the basement membrane of the seminiferous tubule and divide mitotically to produce more spermatogonia and primary spermatocytes. The spermatogonia remain in the basal compartment, while the spermatocytes are located in the adluminal compartment. Primary spermatocytes are located in the middle region of the seminiferous tubule, within the adluminal compartment. These cells have a prolonged prophase that gives rise to the first meiotic division. Secondary spermatocytes are the product of the first meiotic division. These have 23 pairs of chromatids. And this stage is very short-lived and ends with the second meiotic division. Spermatids are the haploid products of meiosis. They remain connected to one another by cytoplasmic bridges, and these bridges result from incomplete cytokinesis and allow for synchronous maturation. So now we're going to talk about the hormonal control of this process. The hormonal control of spermatogenesis follows the general pattern described previously. So this is going to be the hypothalamus produces the GnRH, which promotes the release of LH and FSH from the anterior pituitary gland. FSH and LH in turn stimulate the testes. The gonadotropins were named originally for their effect on the female ovary, but the same names have been retained when we're talking about the male. So the GnRH release is pulsatile, so that means it peaks every 1.5 hours, and LH release follows this same pattern. FSH levels are not as obviously related to GnRH secretion because FSH secretion is also influenced by inhibin and activin. So the FSH targets Sertoli cells. And unlike oocytes, male germ cells do not have FSH receptors. Instead, the follicle-stimulating hormone stimulates Sertoli synthesis of paracrine molecules needed for spermatogonia mitosis and spermatogenesis. In addition, FSH stimulates production of androgen binding protein and inhibin. The primary target of LH is the interstitial cells, which produce testosterone. In turn, testosterone feeds back to inhibit LH and GnRH release. Testosterone is essential for spermatogenesis, but its actions appear to be mediated by the Sertoli cells, which have androgen receptors. Spermatocytes lack androgen receptors and cannot respond directly to the testosterone. So as we talked about here, spermatogenesis is a very difficult process to study in vivo or in vitro, and the available animal models may not accurately reflect the situation in the human testes. For these reasons, it may be some time before we can say with certainty how the testosterone and the follicle-stimulating hormone regulate this process of spermatogenesis. So here I have a diagram kind of showing the visual representation of what I just talked about, the hormonal control in the germinal epithelium. So here we have the hypothalamus, which releases the GnRH, which then is taken up by the anterior pituitary, which then releases the follicle-stimulating hormone and the LH to the testes. And as you can see, the LH then goes through the interstitial cells, which then release the testosterone, which goes to the body for the secondary effects. But if you follow the path of the FSH, those then go to the Sertoli cells, which then release, you see then the release of the inhibin, and they go to the developing sperm, as well as then going to the androgen binding protein, ABP, which then you also see linking up with the testosterone again. So as we mentioned before, this is all from looking at different animal models, and we have not been able to very clearly study the spermatogenesis in human testes. So while this is as accurate as we can get, it is not with complete certainty how spermatogenesis itself works in the human body. So thank you.